Hello everyone. Today we will learn about bed file format that stores the coordinate information of genomic features. Let's take an example of a DNA sequence in the chromosome 1 for an hypothetical organism. There are two types of methods to assign the coordinates of nucleotide bases in a DNA sequence. One is one-based method, another one is zero-based method. In this tutorial, I will show zero-based coordinate method. In the zero-based method, first position of the DNA sequence is considered as 0. The second position has the coordinate of 1, third position has the coordinate of 2, and so on and so forth. This is a 41 base pair length of DNA sequence. The end base, the final base T, has the coordinate position of 40. So let's consider there is a gene 1 in this DNA sequence. It starts from the sixth base of the DNA sequence, so it has coordinate of 5 since it's a zero-based coordinate method, and it has an end position at the coordinate position 16. So we can see here the end position is one base more than the original end position. This is by convention in the zero-based method. One base is additionally considered as the end position. Now we will create a bed file. Bed file is a text file that has multiple columns. Each column contains the specific information of the genomic features. For example, here in the first column, we have wrote down CHR1, that means chromosome 1, since this gene is located in the chromosome 1. And the second column is the start position, and third column is the end position, and the fourth column is the name of the genomic feature. In this example, it is gene 1, and the fifth column contains a numeric value for scores. We can add different values that is related to this gene feature 1. For example, here I wrote down the length of the gene, that is we have obtained from difference between the start and end position of gene 1, that is 11. And the sixth column is the strand information. We can see this gene is in the forward strand or in the positive strand from 5 prime to 3 prime, so the strand is positive. Let's consider there is another gene 2 in this DNA sequence, which has starting position 24 and end position 38. So in the same bed file, we will add this information in a new line. Chromosome 1, since this is also in the chromosome 1, start position, end position, name is gene 2, and length here is 14, and this is also in the positive strand. In the bed file, we stored the information of genomic features in separate lines. That means each line in the bed file corresponds to different genomic features. Bed file can have minimum of 3 columns consisting of first 3 columns, chromosome start and end position. And bed file can have up to 12 different columns that contains the detailed annotation of each of the genomic features. Let's consider another scenario where gene 3 is located in the negative strand. So the question is how we will determine the coordinate position of a gene that is located in the negative strand. Bed file format has a simple solution for that. For a feature that is in the negative strand, it takes the corresponding coordinate position of the forward strand. In this case, for gene 3, the start position will be 12 and end position 24. Here in the bed file, we have added a third line with the start and end position for gene 3. The only difference is we have added a negative sign to indicate that this gene 3 is located in the negative strand. So for a gene that is in the positive strand or negative strand, in the bed file, the difference will be in the sixth column with a positive or negative sign respectively. Let's summarize what we have learned so far. In the chromosome 1, for this DNA sequence, there are 3 genes, gene 1, gene 3, and gene 2. Here, we have sorted the previous bed file. Sorting in bed file means each line in a bed file is sorted in a way so that it is sorted first by chromosome number, then by start position, and then by end position. In other words, we can tell We sort the genomic features from left to right based on their genomic coordinate positions. In the sorted bed file, we have stored the lines from left to right, gene 1 first, then gene 3, and later gene 2. We can see here between gene 1 and gene 3, there are 4 nucleotide overlap. Also between gene 1 and gene 2, the distance is 8 nucleotides. So there are some tools that we can use to retrieve those relationships between different genomic features. For example, using those tools, we can easily get how many bases are overlapping between two genomic features and whether some feature is upstream or downstream of another feature, or what is the distance between features. Also, if we want to merge two features together, we can also use those tools. There are many other operations we can perform on bed files using different tools. Here I have listed a couple of bioinformatics tools that can be used to analyze bed files. One of the most popular tools is Bed Tools. It has a Python wrapper by Bed Tools. Bedops could be a good alternatives. There are a couple of other tools that is available in the public domain. At the end of this video, I also want to touch on BedGraph format, which is based on the bed file. BedGraph file format allows display of feature values in the track format. BedGraph file is very similar to bed file. First three columns are identical between bed file and BedGraph file. But the fourth column is the score column in BedGraph file. BedGraph file also has a mandatory track line where the type need to be specified as BedGraph. Other parameters are optional. We can add name, description, priority. In addition, we can add some other parameters to customize the visualization of the tracks. Let me know your thoughts in the comments below. Thank you for watching this video. See you in the next one.